All right, let's continue. Um, okay. Okay. So we talk about, during development already. Now, what about the opposite? So if aging brain, degeneration of the nervous system. So before we're talking about that, so I would like to maybe emphasize a little bit more about, the synaptic plasticity, right? So in adult brain. So we talk about these a little bit when we talk about learning and memory, right? So one thing to emphasize is in adult brain, of course, you don't have the ability to create the new neurons as much as when you are young, right? As much as when you during the brain development. But throughout the life, you have the ability to change the strength of the connections of the synapse, right? So, and that is underlying mechanism of learning and memory, right? So you can either change the effectiveness of synapse, right? Changing the strength and numbers of synapse, or you can even change the structure, right? You can create new spines, new axons, right? New branches in some cases, right? Emphasize in some cases, right? And even some new neurons, right? Rarely, but possible. Okay. But mostly, most importantly, you change the strength and numbers of the synapse or the connections, right? And that is called synaptic plasticity. And we mentioned this word, right? Long-term potentiation. This is a process that, if you remember, enhance the synaptic strength, right? Following high frequency stimuli. So this is very important for learning and memory, and this is also underlying use dependent change, right, of the synaptic strength. So if let's say you learn something and then like remember the numbers that we talk about, right? So if you try to remember some numbers, but then you never use it again, then you just forget about it. But then if you remember, use it again and again and again, like your ID number, then it's use dependent, right? So synaptic connections become very strong, right? So that is potentiate, okay? And again, mechanism is either you increase the numbers of receptors, right, that are inserted to the surface of the membrane, so more amount of receptors, and also, for the late state, you even increase more numbers of the dendritic spines, right? So that's what happened. And in contrast, if you do not use any information for a long time, those process will be called long-term depressions, right? So you have the all of these basically reverse, right? So those connections or the spines can be removed, okay? And a little bit about adult neurogenesis that you might have heard already. Okay, so this is the evidence in mouse, a lot of the time in rodent brain, that the people agree that the neuronal stem cells still exist in adult, especially in the hippocampus areas, right? And if you remember, hippocampal areas or hippocampus are important for, you know, changing the short-term memory into long-term memory, right? So memory consolidation, very important. So these area have some stem cell hidden, and then once in a while these stem cell can migrate and then become differentiate and become mature neurons, right? And then you can see in these pictures, right, becoming new neurons, having axon and having connections, right? But that is only, like strongly suggested in animal model. Okay, in some other areas as well of the brain, for example, some cortical areas in the rodent, that people suggest that there's some adult neural stem cells as well. And in human, they also find most of the study are, study in the post-mortem brain, right? And they also found that in the hippocampus, there are also some new neurons that can happen to some neural stem cell in hippocampal area as well. And this is kind of a big field also, especially using the rodent mouse model or mouse model. Questions that are usually asked is what factors, or what compound or any conditions that can increase the doubt neurogenesis, right? Because with the hope that, oh, I can make new neurons, right? Anyone want to guess what condition that has been shown and, a lot of people are, agree that, oh, this is, they get a lot of the data in the same directions that it increase new adult neurogenesis. Any condition that increase adult neurogenesis? What condition or what factor? Anyone want to guess? Hmm?
Autism?
Increase. Increase. Which is a good thing, right? So making new neurons in the adult brain, because usually in adult brain, it's like your numbers of neurons are fixed, right? And this is some kind of like new theory that, oh, maybe you can make new neurons, but in what conditions? Anyone?
The the
Ah,
the the autism condition.
the what? Say again. No,
Uh,
in this case, it's like, let's imagine like you guys, everyone, like every one of us, we have these new stem cells hidden in our hippocampus, okay? And which conditions will help to for these stem cells to replenish, to become like, new neurons if needed. It,
uh,
uh-huh.
more stimulated exercise.
Good, yeah. So one of the first thing that, a lot of people studies is exercise, right? So exercise and like enriched environment, for example, um, like let's say if you do this study in the animal, so you can compare between the mouse that are just like do not moving, and then the other one you kind of like put the toy, put the wheel inside the cage, and they move all the time, they're running all the time. So those mouse will have, the one that are active and have enriched environment, will have more rates of neurogenesis, so new neurons developed. What about the opposite? What condition? Decreased adult neurogenesis.
Stress and not enough sleep.
Yes, exactly. Well, stress is well known. Okay, stress is well known. Another condition that is also known from the studies are high fat diet, right? So high fat diet is like it's proven to be not very good result on the adult neurogenesis. Okay, so a little bit of information, but it's not a lot. Still, it's not a lot of percentage that happen in the brains, right? And it's still under a lot of study now. Okay, so before we talk about degeneration, we have to look at normal changes in the brain of the elderly people, right? It's normal to have reductions of the sensory functions, right? Because the hearing, taste, smell can be decreased, right? Especially high frequency hearing. Taste receptors, smell, right? Olfactory receptor is decreased. We talk about it already, right? So everything is kind of like slowly degenerated, but this is normal. So we just have to understand them that, okay, this is normal sensory reduction, right? And visual transparency of the optical light path, So, the lens can become more, it's not as clear as before. But now we are lucky that, we have the lens replacement. So, those that have problems with the lens can change the lens, as it's almost like new lens. Okay. And behavioral change, sometimes they can have reduced short-term memory. They forgot, oh, where did I put my key, right? What did I just say, right? It happen all the time. So that's normal, right? Normal in aging. And sometimes they just like have like a small, bout of forgetfulness. Sometimes they seem to be confused, and they can't really make decision as sharp, as fast as before. The learning rate is slower, right? If you teach them to do something, using like some new program or stuff like that, they probably have a hard time learning, right? Compared to you guys, like, okay, you see it one time and then ta-da, you get, all the techniques and everything. So they have the, slower learning rate, and response to stress is also less ability. But it doesn't mean that the intelligence is decreased, right? That doesn't mean that the knowledge is less, right? Because those are long-term memory, right? And skill and experience. Those are maintained in their brains, right? So a lot of the time, like old people are really smart, right? They have a lot of knowledge, they're really smart, they have a lot of experience. So those still remain, okay? It's just like some of these, new learning skill that can happen. So this is normal change. So it's not like everyone, like if you see your grandma or your grandparents like forget something and you say, oh, you have Alzheimer. No, right? This is normal. Okay. Now, another things that can be changed inside the brain also is the brain weight and volume are also decreased in aging, and the number of the neurons can be decreased by time. It's normal to see, a little bit of shrinkage of the brain, right? But not a lot, right? Just a little bit, right? About like 15% decrease, and this result in decreased number of neurons and size of the dendrites, spine numbers, vesicle numbers, um, neurotransmitters can be also decreased, right, in aging. But again, this is normal aging. But if they have some special conditions, right? Some specific problems. For example, there can be a lot of diseases that are um, considered neurodegenerative disease, but one of the common one is Alzheimer's disease, which is one type of dementia, right? There are several types of dementia, but this is one of the severe type and most common one. And so Alzheimer's, this is the symptoms, major symptoms is memory loss, definitely. And most of the time it's retrograde, right? Amnesia, if you remember. They lost the memory that they used to remember, right? From before. For example, they forget who their kids are, they forget, um, how to perform like easy tasks, like tying the shoelace, right? That's one of the common examples. So those memory that they used to remember very well, right? So those memories are lost, and a lot of confusions happen, a lot of communications problems, speech problems, mood also change, mood swing, depressed, or sometimes aggressive. Restlessness, impaired body function, motor functions also disrupted, and judgment, like big errors, and also like cannot learn anything at all. So this is like major symptoms, right? Basically severe symptoms. And usually, again, see if you look in the MRI, the brain image, and you can see that the brain is like significantly shrink, right? So it is atrophy, or shrink compared to the normal brain. Okay. And the hallmark of Alzheimer's disease is so in other type of dementia, there's really like no significant hallmarks, right? But in this Alzheimer's disease, you can have two major types of hallmark. One is the amyloid plaque, right? And the other one is the neurofibrillary tangle. Okay, the amyloid plaque is the one that you should know because it's most common hallmark of the Alzheimer's disease. But usually, you have to diagnose based on the symptoms, right? Because this is in the brain. Even though people try to find antibody to detect this in the blood or CNFs, cerebrospinal fluid of the patients, it's still not very accurate. Okay, most of the time they will find this after the patient die, right, in the brain, and then they will, they can like make a clear diagnosis that okay, this is Alzheimer's disease. But most of the time, the symptoms will tell, right? So the areas that are affected is the forebrain, amygdala, hippocampus, cortical areas. Okay, so it's result in all of these symptoms. And so the question is, how do we study? Let's say if you want to study Alzheimer's disease or some kind of other neurodegenerative disease, especially this Alzheimer's disease. How do you study? If either you want to study the mechanism or you want to develop some new drugs or new compounds, right? So you can choose again. You usually start from the cellular model, right? Before you jump into the animal model. So cellular model, you can choose between using the cell culture, right? Again, you can choose the neuronal cell line, right? Or immortalized cell line or neuronal cell line, that the one that you can keep from proliferating. Or the best, like I said before, primary neurons, right? So because the primary neuron is the best model in terms of functional, right? Functional similarity to the real neurons because it's derived directly from the mouse brain, right? So you can use these, and a lot of people use these. And or you can use primary skin fibroblasts. In this case, this is the iPSC, right? And we will talk a little bit about this. You derive this into the neuronal cells. Even though it's hard to develop mature functional neurons, but you can study some of genetic information from these model. Organotypic culture is basically, for example, if you cut the brain into the brain slice and you study the whole brain slice instead of separating individual neurons, okay? And you can trigger the model by if directly because the tangle that I mentioned earlier is you can think about tau proteins, right? So beta amyloid and tau are pretty much the most important two hallmark of the Alzheimer's disease, right? So people have these compounds and then you can add these compounds into your whatever model, right? This is the outside model, cell culture model. So you can add this beta amyloid and then, add it into this culture and observe the response, right? And test your compound in the same time with or without your compound, right? Or sometimes oxidative stress can be also a trigger for a lot of neurodegenerative diseases, right? Oxidative stress, free radical, those kind of things. So you can induce also in some model of neurodegenerative disease, you can induce the death of the neurons using oxidative stress as well. Okay, so that's a common simple model. For example, this is an example of neuroblastoma cells, right? It's the first immortal cells that we mentioned. And you might have heard these cells, SY5Y, a lot of very common human neuroblastoma cell. So control, right? And then you treat with beta amyloid, and then you can see, and you use some kind of reactions, chemical reactions to see if it's green. If you see a lot of greens in the cells, that means it's produce a lot of free radical, right? Reactive oxygen species formations or ROS, right? Or free radical. So in this case, with the beta amyloid treated, you have a lot of free radical, right? In the same time, you can observe the damage of the nuclei, damage of the DNA, right? So in this case, you have condensed and fragmented nuclei. That means that inside a nucleus, the DNA is also damaged too, right? In those cells. So this is example of how to use the meta-amyloid. And this is the primary neuron. So you can see the difference, right? In the immortalized cells, you see like you have the cells, you have a little bit of short branches, but it's nothing like the primary neurons, right? In this case, you have long, axons, you can have, these some, you have to make it really mature to have connections. But in this case, you might just see like very long neurons, and you can label it with cytoskeleton marker. And again, you add beta amyloid, which is known to be like a toxin, like a hallmark of Alzheimer's disease, beta amyloid aggregations. So when you add it into the cell culture, then all the skeleton is damaged, and then the neuronal apoptosis is induced, right? So this is example. And then if you have some compounds or some drugs, right, you can add it and then see if the drug prevent or help relieve the toxicity, decrease the toxicity from beta amyloid, for example. Okay. Again, these are example of the cell study in the cell, right? Which is a good first step to screen, right? What is the potential compounds or anything interesting, right? But then let's say if you come up with some compounds that are working, you're not gonna go directly into human, right? So you have to go study that in animal first, right? So the next step is to use animal, or sometimes people skip the cells, and because some people would just argue that, oh, the cell doesn't have anything, right? The same as in the brain, right? So they just use the animal model right away. It's possible too. So animal model for Alzheimer's disease is similar idea. We know the hallmark of Alzheimer's is beta amyloid plaque, right? And neurofibrillary tangle that cause from tau, right? Tau proteins. So we introduce these two into the brain of the mice, right? So APP is the gene that will eventually produce the beta amyloid. So you introduce this in the mice, the mutated version of the APP, and you can see that it will accumulate on the cortex. Tau also, you introduce some of the mutations of the tau into the mice. And both models, sometimes people combine together, APP/tau, right? In that case, you can detect the brain of the Alzheimer, you can detect a lot of, beta amyloid plaque, right? And tau, also you can detect a lot of neurofibrillary tangle, right? That cause, death of the neurons. And then after you make the Alzheimer model, right? Or sometimes you can buy, right? You don't have to make it yourself nowadays. They have like a Alzheimer's mouse, right? You can just buy the mouse and then test your compound, whether it helps with their memory, right? So how do you test their memory? We look at one example, right? Water maze test, where you put the mouse in the swimming pool, right? And then normal mouse will find the platform with no time, right? After the repetitive training, like 10 times. But the mutant mice, MT is mutant. The mutant mice is the mice that you introduced, the tau or the beta amyloid, and it will spend longer time compared to the control mice, right? And here you can introduce your compound and see if it's reverse or it's prevent these confusions in a mutant. Okay, some of the tests you can do like 2 arms, see if the mice remember where you put the food, if it's come directly to the food in this arm, or it's just kind of confused and go everywhere, right, to the other arms. And this is even more complicated, right? You put food on one and then you see how often that it's explore. Usually it's just explore like once, right? And it will just go to the one with the food. But if it's confused, it's just going to go everywhere again and again and again, right? This last one, it's to test if the mouse can recognize the subjects, right? Usually the mouse will tend to be interested in new subject, right? So if you put, let's say, if the green one is the old subject, if the one that they know already, and then you put the new subject here, they will be very curious and they will spend more time exploring new subject, right? But the Alzheimer mice, they can differentiate. They don't remember which one is new, which one is old. So they will spend about the same time for the two subjects. Okay. So this is like a recognition test, right? So there are a lot of behavioral tests that you can pick, right, to study the, in Alzheimer model. Okay, so another disease that is quite common also in neurodegenerative disorders is Parkinson's, right? And this is the problems with dopamine level, right? Everyone know the dopamine level. And then one of the key symptoms is resting tremor, right? Resting tremor, we talked about this already. And so how do you study Parkinson's? Again, you use mouse model, okay? And a lot of the time you inject the mouse with the toxin called MPTP, which is known to damage the substantia nigra, the area that produce the dopamine, mimic the Parkinson's symptoms, right? So if you look at this video, they will kind of like shake, right? And also you test for coordination. This is called rotarod, yeah. Normal mice will be able to stand to hold there. Also a little bit chicken, like the movement is not smooth, right? So that's the normal, that's the Parkinson's model, right? After your injections. And let's see if you want to study, the treatments, like you have some new compounds and you want to know, oh, whether these help, right? You can inject them with the treatment and see the effect. So on the left here is before treatment, right? So it's the PD model, PD mice, which is, this mice is injected with that toxin and PTP, right? And see what happened. Okay, look again. Kind of like lost the coordinations, right? So kind of like fall, whoop, whoop, right away. Okay. And then after the treatment, so if he's improved, you should be able to see something like this. Right? So you can smoothly coordinate and then move his way through, right? So this is after treatment. Okay, so that's just, a two like major example of, the common degenerative diseases. Okay, of course, there are much more to explore, right? And I'll talk about that later. So, continue with regenerations, right? We have maybe a few slides left. I'm not going to talk too much about the stem cells, but regenerations, so this is very important also, right? So the question is between CNS, right? Central nervous system. If you remember, it's include brain and spinal cord, right? And PNS or peripheral nervous system. This includes sensory nerve or motor nerve, right? These two type of injury, what's the difference, right? If you have the problems, if you have injury in the brain, can you fix it? Can you regrow the axon, right? Or versus if you have like, let's say the patients have problem with the finger nerve, they have accidentally cut the fingers, right? And what happened in that case, or crushed the sensory nerve. So in the brain, you can have the injury, several types of injury, right? Traumatic brain injury, sometimes. So this is the result. This picture is the blood, right? So sometimes if the patients come with like a skull fracture, they fracture their skull, then the bleeding, they have a huge bleeding, right? And the blood is clot here, right? Or sometimes they don't have the skull fracture, but they have like very strong impact, right? they fall and then they have like a strong impact. So the blood vessel inside the brain is broken, right? So the whole area here, it's filled with blood clot, right? We call these hematoma, right? So it's different types. So sometimes it's like localized in one area, and sometimes it's like spread through this one. And sometimes you have like a, penetrating injury also. So one of the dangerous thing is that if you see like here, the midline, so this is indicating the severity, right, of the injury. If usually the midline should be in the middle, right, straight in the middle, but because you have like blood clot on one side and it's shift the midline, so these indicate that the pressure in this side is really increased, right? So the overall pressure in the brain is it's increased, and this will be very dangerous conditions, right? Because if you imagine, um, if the patients does not receive the treatment or surgery in time, they the cause of death in most of these patients is because of intra cranial pressure, or the pressure inside the skull is increased, and those pressure that is increased will then push the brain stem. And if you remember, the brain stem contain a lot of regions that are important for, functions, respiratory functions, for example, right? So that's why a lot of cost of death comes from that intracranial pressure. So anyway, just to show you different types, and brain tumor, same thing, right? So when you have increase of the mass, you can also increase pressure in the brain as well. And in this case, again, push the midline as well. Stroke also, another type of problems that can cause damage in the brain as well. And so one fact about the brain is that, if you're missing oxygen for like 5 minutes, some of the cells, brain cells, are already dead, right? And you know that if you have damage in the neuronal cells, if the neurons die, you can't it's really hard to regenerate, right? It's almost impossible to regenerate, right? So stroke, you have like three major types, right? You have two major types: ischemic or hemorrhagic, right? Hemorrhagic stroke is a stroke that the patients bleed, right? The blood vessel is broken, and then the bloods come out in specific areas of the brains, right? And ischemic strokes, blood vessel is still intact. However, there's something blocking that blood vessel. That's why you have some area of the brain cannot receive enough blood. And it's further divided into thrombotic stroke and embolic stroke, right? Thrombotic stroke is when the blood vessel is, thickened, and you have, a lot of these plaque on the wall of blood vessel, and some small thrombus can block it, right? So you block the blood flow to the brain. Embolic stroke is that you have some kind of plaque somewhere else in your body, for example, in the heart, vessel in your heart, and then it flow. Sometimes those blood clot fall off, and it become these embolus, and then it go up to the brain and block the small blood vessels in your brain, so causing the stroke as well. So you can imagine, if this is blocked, the area behind these regions doesn't have any blood, right? And your blood, you know that the blood deliver oxygen, right? Oxygen is needed for the neuronal cells to make the energy and maintain, the neuronal cell activity, right? Without oxygen, they die. Okay, so this is the different types of the stroke. Now, what happened after the stroke or the traumatic brain injury? You have first primary damage, right? So during the injury or during stroke, you can have neuronal damage directly, right? Or sometimes you have blood-brain barrier breakdown, okay? Or blood spinal cord barrier, sorry, blood spinal cord barrier breakdown, depending on what type of damage. Sometimes you have the necrosis, right? And that's primary damage, which is the damage that happened right away during injury. But that is not as severe. In some cases, the worst come from secondary damage, right? Which is the damage that occur after the first event, right? Which can happen, a few days after or a few hours after, right? If the primary cause is not treated properly, right? Secondary damage include inflammation, excitotoxicity. That means that some neurons if it's damaged, it's going to release a lot of excitatory neurotransmitter, which is glutamate, right? If you remember. So it's going to cause toxicity in the brain, right? Ischemia, free radical, apoptotic edema can also be secondary problems, right? So now first question is, if you damage the axon, right? Let's say you have the injury, you have the traumatic brain injury, you cut the axon, can it be regenerated? And the answer is depends, right? If the damage occur in the peripheral nerve, right? For example, the sensory or motor nerve, it can be regenerated easier than the CNS nerve, okay, or the nerve in brain and spinal cord. And these are the reason for that, right? So in the peripheral nerves, axons have the macrophage, right? So the macrophage can effectively clear the myelin debris, right? So you have the axon and the axon is covered with myelin sheet, right? So in the peripheral nerve, you have macrophage locating around and it's remove myelin debris very easily. Second reason is that they have the Schwann cells, right? The Schwann cell is the cells that make myelin sheath for peripheral nerve, right? Schwann cell can release the growth promoting signal, okay? And the neuron itself, the property of peripheral neuron itself, it's able to express growth related genes, okay? So it has the ability to re-express whatever the genes that they express during development for the PNS, okay? In contrast, if you look at the CNS or the neurons in the brain or spinal cord, they don't have macrophage, right? They have microglia, right? Immune cells, microglia, but the microglia it's not as effective as macrophage, so it's less effective to clear that myelin debris. Another reason is they have the astrocyte, right? Astrocyte form the scar to basically block the growth of the new axon. And also they don't have a lot of tracks for the axon to grow as well. Imagine the brain, like everything is packed, so they don't have a lot of good tracks for the axon to grow. And also, most important reason, these astrocyte and oligodendrocyte can release inhibitory signal, okay? And when the inhibitory signal is released, it's blocked the regrowth of the new axons, okay? So that's the reason why when the patients have damage in the brain and in their spinal cord, it's so hard to repair compared to if they have, problems with the, hand, the limb, nerve, for example. Those can be easily repaired. But what can happen in the brain if, after the patients have the traumatic brain injury is not, sometimes they can recover, right? If not fully recover, but some level, right? And those recover come from cortical reorganizations, okay? And it's underlying functional recovery, okay? But how do you make functional recovery? Most importantly, you have to stimulate the brain, right, by using the physical therapy. For example, if you have the problems with the hand regions, right, with the stroke, that the patients have problem with movement on the other side of the body, right? So you do physical therapy to kind of activate the cortical reorganizations, right? So what is cortical reorganizations? Let's see. So the brain, even though the neuron doesn't have ability to regrow the axon, right? For example, in this region, it's just one area of the motor area of the brain, right? This area, zoom in from these. The motor areas of the hand, movement of the hand. And then this area is where the stroke happened, right, in the beginning. So after the injury, after the rehabilitation, this area, it's gone, right? So the activity of this area is decreased because, you have the stroke in this area. So the activity is decreased, but other area increase its activity. You can see black mean high activity, right? So other areas becoming, compensate and then perform, being more active. So you recruit the um, other areas to help with the movement of the hand. Basically, you recruit, maybe the arms, you move the arms more or something like that after the physical therapy. Okay, so the pattern of the cortex or cortical areas that change after the injuries, it's called cortical reorganizations. Okay, and this is not because of the new neurons, this is not because of the axon regrowth, but is the reorganizations of the existing neurons, right? The healthy neurons become compensating, okay, with the injury, right? So a little bit of, the fact about injury and regenerations. So how do you study these regenerations? What's the questions? Of course, the questions usually is, how can we help, right? Treating the patients that comes with the brain injury, spinal cord injury, right? For example. So the first thing that we have to realize is that, again, remember to decrease, to protect the further damage, right? By secondary injury or secondary damage. Okay. So because otherwise, if you don't protect secondary damage, you're going to have increased the further death, right? So how do you prevent secondary damage? People try to develop anti-apoptotic molecules, anti-inflammatory molecule, free radical scavenger, getting rid of the free radical, a lot of things. And trophic factor, also something that we currently use in clinic as well, even though it's quite expensive. But trophic factor is, all a mixture of like growth factor that can help maintain, the neuronal health, prevent it from secondary injury. And also, a lot of research is trying to increase the regenerations of the neurons, right? In the brains, for example, trying to increase plasticity, more supporting more reorganizations of that neuronal circuit. And in spinal cord, trying to increase growth, axonal growth, okay? How? Increased plasticity, again, trophic support, some of the trophic factor, growth factor has been studied, and they are trying to enhance neurogenesis also. Is another big field, right? In spinal cord, we learned that the reason why the spinal cord or central nervous system neurons cannot regenerate very well, it's because of inhibitory signal, right? So a lot of research try to develop some compounds that decrease inhibitory components that are released from the glia cells, from astrocytes, from oligodendrocytes, okay? And they also try to, develop some physical guidance channel, and then adding some cues to help regenerating the spinal cord. How do you study spinal cord injury? So unfortunately, if you do animal model, you really have to crush you have two ways. You can use something sharp to completely cut the spinal cord, or sometimes, people want to mimic the spinal cord injury in human, which is, sometimes comes from accidents. So, you have this kind of like a huge some kind of pressure crush the spinal cord, right? So it's not cut, but it's crushed, right? So damage the spinal cord. And so and then they test the compound, so on. So sometimes it's just it's too bad that you have to injure the mice. In this case, of course, they will, more or less have the painful experience, right? After the operations. So it's always good to study in the primary neurons first. This is another good example. Why do you need to look at, potentials of those compounds in the primary neurons first? So in this case, again, using primary neurons, right? Isolated from the mouse. And so you put the neurons in one side and then you allow the axon to grow onto another side, right? And then you induce the injury in the middle, right? You induce the injury in the middle. And by, you can push some kind of high pressure to damage this axon. And then you can study what compound can facilitate the axon growth, for example. Okay, so that's one of the model that you can avoid using animal model. Okay, so what happened to patients with spinal cord injury? So the problem is they lost connections, right, between brain and muscle, because we learned already in motor system that brain send a signal, upper motor neuron, down to spinal cord, right? And then spinal cord send a signal to the muscle. So if any level of the spinal cord is, injured, then you disrupt that connections. So how do you fix that? You reconnect that connections, right, between the brain and muscle, right? So basically you bypass the spinal cord. And so, for example, in this case, you have brains, you have the spinal cord. If this is the area of injury, then this muscle will not be able to function, right? Because there's like blockage or broken connections here. So a lot of study trying to, insert like a implant the trigger to the brain area. So this this area will give the signal, right? Okay, which muscle to be moved, right? And then you interpret the signal and then stimulate directly this spinal cord section, right? That control this muscle, wireless, right? So this can help, bypass the areas of the injury, okay, and help improving motor, right? So by the way, so again, all of these, that I picked and show here is just to trigger, your brain and your thought a little bit. If you are interested more in detail, of course, nowadays you can literature search, right? And then look more into details. So this is similar idea with the human brain also. You have the signal in the brain, which now you can detect, right? You have the chip implant and then you can encode, right? Okay, what these patients want, and then bypass the spinal cord and stimulate also in this case directly, right? The arms directly also it's possible. There's another, study in spinal cord nowadays and that is like very interesting. Reading the signal from the brain and then bypass it spinal cord to trigger the muscle movement. Okay. Another field is the hope to use the stem cell to treat for the cell therapy for spinal cord.
Oh.
Any questions? Okay, so the idea in this case is if you have the spinal cord injury, right? So hopefully you can get some patient skin, right? Make it into iPSC, right? You know about iPSC, induced pluripotent stem cell, right? And then you can hopefully develop them into precursor cells, make it into, sphere and then introduce it into spinal cord, right? And then it fix, it grow into the new neurons. So, okay, before we continue, I'm gonna keep the quiz for later. Let's continue first, okay? Let's talk about stem cell. Since you guys learned about this already, it will be quick, okay? So this slides, I think I put it in a handout. You can just look by yourself. This is different source of the neural stem cells, embryonic, or you can get it from skin cells, right? Induced into iPSC, and then you can differentiate it into neural stem cells and then eventually into the neurons, okay? But one thing to keep in mind, let's say if you have neural stem cells, right? It has the potential to become any type of cells in your brain, right? So this is one of the challenge. How do you make sure that it's really changed into 100% into the neurons, okay? And another question is what kind of neurons that you are changing it into? Dopaminergic neurons, glutaminergic neurons, right? acetylcholine, the neurons that produce acetylcholine and so on, right? Because there are so many specific types. That's why the limitation of the stem cell lies here through specific differentiations, right? That's why primary cells give you like a, more functional because this comes from the specific types. So applications can be a lot, right? So basic knowledge can be used as a model, drug development, and use it as the creating some compounds, right? Or transplantations, right? In some cases, it's useful, it's successful for transplantation, but the brain still we haven't get there yet. Okay. The idea, this is just some useful idea. It will be great if it's become real. So the idea is that if, let's say, if you have neurons and you have the Alzheimer, right? You have the plaque, amyloid plaque, or the tangle. And the idea is that if you have stem cells that can basically release growth factor in these regions, then it will help, the neurons to create some new neurons. And then also any compounds, right, that can restore endogenous neurogenesis, right? Like we're talking about, like, and then increase the neuron to replace those, cells that are dead. That would be the hope, right? But still, nowadays, there's still not just such a compound, right? But it's a good idea. Parkinson disease as well. So this is normal brain, right? So you have substantia nigra, which send dopamine into basal ganglia, right? One of the areas. Um, if in the Parkinson disease, this cell die, right? So the hope is, if you have the neural stem cells that can protect the rest of the neurons, at least you can protect further, death from these dopaminergic cells. Or even better, if you can, put the dopaminergic precursor cells here, and then hopefully it will become mature neurons and then produce dopamine directly in these regions. Okay, so that can be another hope as well. And you can, again, different source of the stem cell, fetus, embryonic, iPSC, or even the bank, right? So this is just the idea for the hope. This is another useful, a proof of useful. This is also a while ago, and it's one of the, in the beginning that we use iPSC to study Alzheimer model, right? So the idea in this case, you get the skin from the patients, right? Fibroblast, and you, reprogram, make the iPSC, and then change it to neuronal progenitor cells and differentiate into neurons. And then you take these neurons and study, whether the cells express, beta amyloid, the mutated version of the APP that create beta amyloid and those and these. And you can use these for, study both genetic background or treatment possibility as well. Okay, so that is possible. Now, the most exciting one, I think, is the use of the stem cells in a spinal cord injury. Now it's still in clinical trial, but it's very interesting progress. So the idea in general is that, so you have the spinal cord injury. So you mix basically the stem cells, right? The stem cells, neural stem cells with a bunch of growth factors. So list here is a bunch of growth factors and some kind of gel to kind of guide, combine these together. And then you inject it into these scar and you observe the regrowth of these neurons, right? And so in the beginning, in the first phase, of course, they did this in rodent model, right? And they look at, so basically they cut, if you look at the graph, so at first, this is in the beginning, after they cut that spinal cord, right? And then they add these neural stem cells, and then they can observe, the axon is actually regrow, right? The stem cells actually regrow successfully, which is the good news. And in addition, they have to observe the symptoms as well, right? So usually spinal cord injury, when you cut the spinal cord, the mouse is having problem walking, right? So he can walk normally. So the pink line is like a normal, it's like a mice without stem cell, right? So the progress is quite slow without the graph or stem cell. But then if you put the graph, this is the walking score. He can walk much better, right? But then, sadly, at one point they have to recheck. But this is really come from, the new neural stem cell. So they have to cut again, transection, cut the spinal cord again, and then the symptoms go down. Okay. So basically, well, it's useful to see that, it's possible to do that in rodent. And I think study after this is also a long time ago, but if you follow the study nowadays, so they move from rodent model into primate model, and then I think now it's already in human clinical study also. Okay, so again, this is just a summary slide on how can you use the stem cells, the hope for use the stem cells, right? So a lot of these applications, okay? All right, so last slide, again, I give the example of just some basic study in this field, in the developmental degenerations, regeneration, neurostem cells. But you can have any other fields of neuroscience that you're interested also. And I want you to spend some time to explore, okay? So that's the reason why I need you to do some short report, okay? So these can be, like one page, just one page, and get a chance to read and, appreciate. Pick any paper, okay? Article, not review, okay? Not review. So pick any article in any field of neuroscience. It can be the field that we talk about today, or it could be any field in neuroscience that you're interested, okay? And then pick that one paper and summarize these 4 points. What is the purpose of the study? What is the main questions of that study? What is the model that they use, okay? And what are major findings? What did they find, right? And take home message after you read this message. What did you get from this paper? Okay, everyone. And then the deadline that I propose here is Sunday, April 20, okay, for the report. All right. And I will need one of you to combine all the reports from everyone and send it to me. Okay, you can send it in Google Drive, or you could create Google Drive or something and then send it to me. So each of you will have to send 2 things. Okay, first is the original paper, right? Original paper. Second is this 1-page summarized. Okay, original paper, you put it, it's usually PDF file, right? And then 1-page summarized, it will be easy if you put it in PDF or Word file, I don't care, right? So make sure that you send it to, I don't know who, so can you tell me now, like who's gonna be the one that can combine all the report? Anyone here can do that?
I think maybe someone can make a Google Drive and then everyone can add their in. Um, and
Okay.
each department would have like a representative.
Okay. And so you guys can coordinate, right? For each, because usually every year I would have like one person that send that final email and final drive to me that include like every single one of you in the class. I don't know if you coordinate, you might have to coordinate between the 3 programs and then come up with like the final person who submit everything to me. Is that possible?
Yes, we can work it out.
Okay. Okay, and are you okay with the deadline? Or you want earlier or later? I'm flexible. I just thought before, I thought that you have the exam on the 19th. But now if you have exam on the 16th, then you have some couple more days to do the report. I don't think it should take too much time. Just to like summarize the paper. So it's
I
okay?
think mine is okay.
All
Uh,
right. Mm-hmm.
this is the report is individually, right? And not
Individual. Yeah, individual. Individual. So each of you have to pick like, different papers. Any questions about, the lectures or about the assignments or about anything at all? About the exams? No? No questions? Okay, so the exams again, I already mentioned it earlier in the course. It will be, I don't know, 16 April 16th. But you should check again, double check again in your schedule. Because I already sent the exam to your academic assistant, okay, and she will probably take care of, how you take the exam and everything, okay, and after I get the exams score from you guys, I'm gonna and also reports. So as soon as I get all the reports on Sunday, April 20, then I can finalize the grades, and then you guys should know your final grade um the week after April 20. And then you guys going out to research, right? To some kind of internship or something, right? After,
yes.
okay. All right. Are you excited?
Scared.
Anyone's going to Thailand? No? We used to have some students come to, when I was there, some students come to Thailand. Yeah, it's a couple, like maybe 4, 5 students, so 1 year before COVID. And then after COVID, I don't know if anyone, coordinate with stuff in Thailand. Do you still have some professor from Mahidol University from Thailand come and teach you?
Yes, yes. A few courses that have
Few classes? Okay. What's the name? You remember?
uh one of them is developmental. Yeah, and the other one is animal physiology.
Animal physiology. Is it Dr. Tep Manat?
Yes, yes.
Oh yeah. Yeah, well, I was like I worked at the same university as him. So he came he came to Hanoi, right? So it's on site, right? Animal
Yeah.
physiology. That's good. That's good. Yeah, he's from the same department. So his expertise is cardio physiology. Yeah. What about Dr. Atit? He used to go there also, but I don't know if he's still going there. Dr. Atit, Dr. Kanit, um, yeah, they're usually teaching about something about biology. So okay. Um, oh, report can be in Word file. Can be in Word file. No, you don't have to write down. You can type it in the Word file. Okay. Any other questions? All right. So during this time, if you have any questions, you can contact me, right, by email. I hope everyone have my email address, right? So that's where I send it to you guys. And otherwise, good luck for the exams and good luck for the intern and have a very nice summer. And again, if you have a chance to come to US, to Chicago, let me know. Okay. So, and again, if you guys are interested to pursue your academic, journey in neuroscience or, any field, related field, and you want recommendation later, feel free to ask me. Okay. Reach out to me and then introduce yourself. Um, tell me who you are a little bit, and I'll be happy to write a letter for you. Okay? All right. So I hope you learned something from this course, and hopefully I'll see some of you guys sometime. Okay? Good luck. All right.
Thank you.
Mm-hmm.
And good night.
Thank you. Have a good weekend also. Good luck with the exam next week. Nail it.